On this slide, we're looking at some images that were taken under a light microscope. Let's start in the upper left-hand side of the slide. You're looking at two identical types of cells in these two images. These are epithelial cells. These are eukaryotic cells. We can see that because of this structure right in the middle. This is a nucleus. Notice that this cell has sort of an unusual shape. It's quite flattened. It doesn't have a nice round, plump, three-dimensional shape. It looks sort of like it's folding in on itself. It's collapsing a little bit. This is an epithelial cell that has aged. It's closer to the upper layers of your skin, for example. It's preparing to be sloughed off. Over here we have 1, 2, 3, maybe 4 epithelial cells, but of course they look quite different than the cell on this side of the image. This image, this cell, has been stained. You can see that it's taken up this sort of a blue color, particularly in the area of the nucleus. Over here, these cells have not been stained. And I think you can see that you just get more detail when you stain a cell. Different stains are intended to adhere to different types of molecules with more or less affinity. So this stain adheres particularly well to the DNA that's sitting in this nucleus, and that's why the nucleus is staining so darkly. Over here, we can still see details. We can still tell that these are individual cells, and you can count the cells because each cell has a nucleus in it. But it's just more difficult to see anything with any clarity when you don't stain a cell. Now let's take a look at the pair of images in the middle of the slide. On the left-hand side, I think you'll agree it's pretty hard to see much of anything, although you are looking at a smear of blood cells over here, white blood cells particularly. Now if you look very carefully, you can see these circles, little circles here. Those are the nuclei of these eukaryotic cells. But boy, it's hard. It's hard to see individual cells over here. And that's because this preparation has not been stained. Now, if you look over here on the right-hand side, you see the exact same smear of white blood cells that has now been stained. Again, these are eukaryotic cells. They have nuclei. They have particularly large nuclei for this particular type of white blood cell. And you can see there's some mass of cells over here that looks quite abnormal. These are actually cancerous cells. And one of the ways that we distinguish cancer cells from other cells is by the fact that they look so abnormal. They stain differently. You can see they're much darker than these other nuclei. You can see that they're a different size, that they're shaped a little bit differently, that they're clustered together. All very abnormal. But I think you'll agree that the staining has brought out a whole lot of detail that we just couldn't see over here. Over on the right-hand side, we have the last pair. Now these are two different images of the same slide. So two different fields of vision on the same slide. These are also cancerous cells. Up here, we have this sort of blob-looking cell with an enormous nucleus. Hard to see a lot of detail here. Down here, we get a whole lot more detail. Let's switch gears here and talk about a couple other types of microscopy that we should be familiar with as students of microbiology. The first type is called fluorescence microscopy. Now, if you recall, light microscopy uses visible light in order to view specimens under the microscope. Fluorescence microscopy extends the range of what is used to view the specimen. Now, visible light is the type of light that ranges from about 400 nanometers to about 700 nanometers in wavelength. I want you to notice that this spectrum of light, this visible light range, falls right here, visible light right here, in this big spectrum of other types of electromagnetic radiation. All of these things have a wavelength. That's what they're showing us up at the top of this image. So whether you're talking about visible light, the little sliver right in the middle, or infrared, or radio waves, or microwaves, or ultraviolet, or X-rays, all of those things are forms of electromagnetic radiation. So when we look up at the top of the image at this wavelength comparison, you can see that over here on the right-hand side, the wavelengths, in other words, the distance between the peak of one wave to the peak of another wave, that's what we call a wavelength. You could also measure what's called the trough, the bottom of one wave to the bottom of another wave to calculate wavelength. The wavelength on this end of the spectrum is quite long. These are long wavelengths. Over on the other end, these are much closer together, these waves, so they have a very short wavelength. This is a spectrum. We go from one extreme all the way to the other. And as we go to shorter and shorter and shorter wavelengths to the left, we're also talking about forms of electromagnetic radiation that have more power, more energy. Look at what's at the extremes. Radio waves. They don't have a lot of energy associated with them. You know that radio waves are not damaging to you, for example. They don't destroy your tissue. They don't penetrate through you and injure you. But compare that to things that are farther over here on the left. X-rays, for example. X-rays can penetrate your tissues. We use them in medicine to take images of the inside of your body. And if we keep going to the left, we hit gamma rays, the kinds of rays that come from, for example, nuclear material. X-rays and gamma rays are powerful enough to penetrate through you and also to damage your cells, where radio waves and things over on this end of the spectrum are not damaging to us. They have a much longer wavelength. So back to fluorescence microscopy. We're going to extend the range of wavelength that the microscope is able to use to help us view a specimen. Fluorescence microscopes not only use the visible range, but also into the ultraviolet on this side and the infrared on this side. So they're capable of a longer range of wavelength that they use in order for us to see the specimen. Why does that matter? What does that do for us as the person who's using the fluorescence microscope? Well, fluorescence microscopes give us a better resolution. Remember, we defined that as the ability of the scope to show us two things that are very, very close together, and we still see them as two separate things. Remember, we talk about resolution in terms of resolving power. And we said that a light microscope has a resolving power of about 200 nanometers. As long as two things are at least 200 nanometers apart, we will see them as two different things. With a fluorescence microscope, those two things can be only 20 nanometers apart, and we will still see them as two different things. So, number one benefit for the fluorescence microscope is it has a better resolution. Now, the reason we refer to this as fluorescence microscopy is because we don't just stain our specimens with ordinary stains when we're doing this type of microscopy. We use fluorescent tag molecules. Now, what I mean by that is certain types of molecules that, again, will bind to materials in a cell. They might bind to protein, or they might bind to DNA or some other type of molecule. But this time, they're also capable of fluorescing under an ultraviolet light. So not only are we going to see a color, we're going to see a very bright fluorescent color when we look underneath the microscope. Now, fluorescence microscopy can be very complex optically speaking, but primarily when we're using it for our microbiology needs, it looks a lot like a form of dark field because the background, the field, will be very dark, black even, and the object will have these very bright fluorescent colors. The other type of microscopy I want us to be familiar with is electron microscopy. You're looking at an electron microscope over here on the right side of the slide. Electron microscopes are enormous, very difficult to use, and very expensive, as you might imagine. Electron microscopes don't use visible light and UV and infrared. They use a beam, a stream of electrons to help us view specimens. So very different technology with an electron microscope. The electron microscope provides us with the best resolution that we're capable of right now in the 21st century. It has a resolving power of less than 0.2 nanometers. So as long as two objects are at least 0.2 nanometers apart, an electron microscope will allow us to see them as two different objects. So that's pretty amazing. That's a very, very, very small number. There are two kinds of electron microscopy. There's what we refer to as scanning EM and what we call transmission EM. The difference between those two is how you're going to see your specimen. Under a scanning electron microscope, the beam of electrons is going to show us the surface details of the object. And I'll show you what I mean by this in a minute. In comparison, under a transmission electron microscope, what we're going to see is the internal details, what's inside of the specimen. Electron microscopes, as I said, are very expensive. If you compare the costs of microscopes, a modern, high-end compound light microscope is going to cost you thousands of dollars, but, less than $10,000. In comparison, a fluorescent microscope is going to cost you tens of thousands of dollars. An electron microscope will go anywhere from hundreds of thousands of dollars into the millions of dollars. So you don't find electron microscopes at very many institutions. They're just too expensive. Now take a look at these amazing images. These were taken under electron microscopes. And what you can see here are 3 images of scanning electron microscope technology. Remember, the scanning EM shows us surface details. Transmission EM shows us the internal workings. Up at the top here on the left-hand side, yes, this is a snowflake, an individual snowflake. And as you can see, I can see the details of the surface of this object very, very clearly. I can't see inside the snowflake, but I can see its surface very, very clearly. Over here, you've got another blood smear. but look at how detailed this is. You can see individual red blood cells. Those are the ones that have that central sort of dimple in them. You can also see white blood cells. You can also see platelets. Amazing, amazing level of detail with an electron microscope. This last image over here, what you're looking at in these pinkish sort of red colors, these are eukaryotic cells that have been grown in a culture dish in the laboratory. And what you're looking at in yellow, this is our friend SARS-CoV-2. This is the coronavirus that is currently plaguing the planet. I want you to notice the size differential. Each one of these little yellow particles, these are viral particles. They are in the process of bursting out of a cell right now. They've been replicated and they're ready to go out and infect other cells. Compare that size to the size of the cell that they are infecting. Viral particles are very, very small, visible only with an electron microscope. The images on this slide were taken with a fluorescent microscope, and I think you can see the colors are pretty remarkable. One of the nice things about fluorescence microscopy is that we can combine the fluorescent tags that we use. So, for example, you can combine a fluorescent molecule that will bind to the membrane of a cell and show this bright red color, while also tagging the cell with a molecule that binds to internal structures, shown in green here. Over here, again, you've got different colors, different tags. You've got blue and you've got green, again, binding to different structural components of the cells. Fluorescence is really amazing. It's very detailed. It's very bright, very obvious, the individual structures that are tagged in the cell. On this final slide, we're looking at a comparison between the level of detail that we would see using a compound light microscope and using electron microscopy. And I like this image because it also shows a comparison between transmission electron microscopy, or what's labeled as TEM, and scanning electron microscopy, or SEM, S-E-M. What we're looking at across the top here, these are yeast cells. These are single-celled fungal organisms. And remember, by our definition, that is a microbe. Fungi can be single-celled or multi-celled. They can be microscopic or macroscopic. We just happen to be looking at a single-celled microscopic form, which is yeast. So under the light microscope, the yeast cells have been stained. They look quite dark against a bright background. So this would be a bright field image. Compare that with yeast cells that are being imaged under a transmission electron microscope. You can see one adult or parent yeast cell here, and it is budding to produce a new cell, a daughter cell. You can also see that over here. You just can't see it in any kind of detail. This cell, for example, right here, has a bud coming off of it. Under the electron microscope, not only can I see all kinds of detail of the parent cell, I can see individual organelles in this parent cell. I can also see internal details in the bud. Now, compare that transmission image with the scanning image. Here's the adult, the parent yeast cell. Here's the bud. Notice that we're not looking at any internal details here. We're looking at the surface. But you can really get a sense of the three-dimensionality of these yeast cells using this type of microscope. Down here, we're looking at a much smaller type of cell. We're looking at bacterial cells. Here's the compound light microscope image. These are bacterial cells that have been stained. You can see them as pink objects, darker objects against a bright background. This is bright field. Now, this happens to be an image that was taken using a high-powered lens. We're looking at these cells under the 100x lens. They're tiny, even under 100x. Now compare that to what we would see using the transmission electron microscope. Much, much more detail seen in each individual cell. And of course, when we look at the SEM image, now we're looking at the surface of these bacterial cells. Really amazing, the amount of detail you get with an electron microscope.